a very narrow view to the things. When bacteria die, we must be a virus, right?
I can just feel the pain of every microbiologist and virologist and biologist in general who might be watching this video. Hi, I'm Dr. Wilson. I'm a PhD molecular biologist, and welcome to another COVID-19 debunking video. Imagine if flat earthers suddenly believed that they were experts in biological sciences. Well, then you'd have the topic of this week's video, virus deniers. These are people who actually believe that viruses can't make us sick because they don't exist. Yeah. Pretty crazy. I'm going to be debunking one of their biggest ringleaders, Stefan Lanka. So strap in, this is going to be a pretty crazy one, but I'm going to be bringing up a lot of cool science.
Yeah, me.
Get ready. From an interview where Thomas Cowan, who I've covered before on this channel, interviews Stefan Lanka. And right in the beginning, he starts talking about viruses for bacteria, or otherwise known as bacteriophages. These are viruses that infect bacteria. And right away, he has some very interesting things to say about these very well-known viruses.
Bacteriophages. Phage from phagocytosis, to eat, to swallow. But those structures are not eating the bacteria. This is a misconception. These are mini spores which can themselves develop again into larger units.
I can just feel the pain of every microbiologist and virologist and biologist in general who might be watching this video. Yes, he did just say that bacteriophages are spores.
Mini spores. These are mini spores. Mini spores.
Yes, he did just say that bacteriophages are spores that will turn into bacteria.
Which can
Will turn into
larger units.
Yes, he did just say that bacteriophages are spores. Turn into bacteria.
Dr. Stefan Lanka says that under certain conditions, the bacteriophages can turn into larger units. Caveat is under certain conditions, just like the bacteriophages themselves are formed only in certain conditions.
Wow.
Dr. Wilson proceeds to educate us that some bacteria, under certain conditions, form spores, as if this is what Dr. Lanka was talking about. But it is not what he is talking about. Dr. Lanka, I'm well sure that he is well aware that bacteria form spores. He is talking about mini spores, and this guy showed in his own video. Look at him, go ahead and show some paper, some writing, some stuff that looks sciencey. See, there's spores here. Dr. Lanka is wrong. See, he's even going so far as to say bacteriophages are so much smaller than bacteria spores.
Wow.
Dr. Lanka is saying they are mini spores.
It is orders of magnitude smaller than a spore or bacteria. It looks completely different. It has its own unique genome. It has its own unique proteins and its own unique structure. And of course, this specific genome is far from capable of coding for an entire new bacteria cell. That is completely wild. Far from capable of coding for an entire new bacteria cell.
Larger units.
Dr. Lanka has simply said under certain conditions, these mini spores can turn into larger units, perhaps something that precedes the bacteria.
The reality is that many bacteriophages actually kill bacteria. This can be easily measured in an experiment called a plaque assay.
At this part in the video, he wants to tell us that he knows bacteriophages infect bacteria cells because when there's a lawn of healthy, chemically fragile bacteria cells, the more bacteriophages you sprinkle on that lawn, the more bacterial cells end up dying, as if it means something. Lanka says the bacteriophages are made by stressed cells, and Dr. Wilson's saying that the bacteria that come into contact with the bacteriophages end up dying and more bacteriophages are found. He's saying that the more bacteriophage we add to these fragile bacterial cells, the more end up dying.
This is an experiment where an isolated bacteriophage is diluted over a series of tubes, and each dilution can be plated onto a plate of full bacteria. If there's a lot of phage present, as you see towards the left-hand side of this image, you get complete lysis, or a complete destruction of the bacterial lawn. And as you dilute the bacteriophage more and more, you see less and less cell death. You can then pick out virus from any of these individual circles where a bacteriophage landed and started killing the bacteria cells and further propagate that one bacteriophage colony.
In other words, we can harvest materials from dying bacteria, put them on healthy bacteria, watch them die, harvest materials that result from their death, put them on more bacteria, watch them die, and the more we put on healthy bacteria, the more healthy bacteria will end up dying.
In the rest of this interview, Stefan Lanka goes through 7 points that he thinks destroys all of virology. And I'm going to address every single one of them.
The first step I mentioned already. Since endless, every biologist believes when his cell cultures are dying in the test tube, right, that there must have been a virus, but they never controlled it. So we controlled it twice already, and we get the cells dying without infectious material.
So this is what you'll hear many Stefan Lanka followers repeating ad nauseum. He is basically saying that if you take an experiment like what I just showed you, a plaque assay with bacteria, and do something similar with human cells, that there's never a control for it, and that the cells will just die anyway, whether or not there's a virus.
It is true. Some people are saying that there is no control for any of the virology culturing experiments. Some people saying there's no control at all.
It's complete baloney.
If Dr. Stefan Lanka is saying there is no control in these experiments ever, I would assume that he is meaning there is no control for the addition of protein packages in the physical proximity of the cells. If you have parvovirus and you are seeing how destructive it is to some dog cells, you should also include human measles virus as a control to see that when this dog cells die after having contacted parvovirus, to see if it's actually just because the cells were contacted by a protein package that shattered their chemistry.
Experiments like this always have a control. Always. Often it's called a mock infection.
Stefan Lanka doesn't mention using other microbes that are theoretically not infective to the cells. He mentions in the interviews using protein packages. I would say use something of the closest chemical compatibility to the microbial particle you're experimenting with.
Here is a great example of this that even goes a step further. This is an example of a paper where they looked at Japanese encephalitis virus and how it infects human cells. This is a picture of a plaque assay, just like how I showed you with bacteria, except with human cells. And what the researchers did was they mutated the virus. So they were able to reduce the size and number of the plaques just by mutating the genome of the specific virus.
In other words, as I see it, we can harvest protein packages that have accumulated as a result of dying cells. We can place them on cell culture dish and observe a certain amount of cellular death as a result. We can change the conditions of the cell culture and harvest the particles that have resulted from those deaths in the cell cultures. When we put the original protein packages that we harvested originally from the dying cells, we can put them on this other cell culture which is in different conditions, and we can notice a different kind of pattern of cellular destruction. We can collect the protein packages that have resulted from that in that different place, and we can place them in another culture of cells and observe the kind of cellular destructive pattern that occurs, and we can go and harvest the protein packages again from that cellular culture that have arisen from the destroyed cellular lawn. And by this stage, when we place those protein packages on our cellular lawn back in the original conditions when we first infected a cellular lawn, the effect might be different because they'll be slightly different. You would rationally expect there to be different kinds of protein packages left over when you introduce the same kind of protein package to the same kinds of cells in different environments. The different environment will cause a different character of breakdown, will turn out a different array of compounds.
And yes, they included mock controls in their studies. What Stefan Lanka is most likely describing in his experiments is his inability to culture cells properly. It's not hard to culture cells properly, but if you don't do it properly, the cells will die. So that's probably what Lanka is doing, and then he's observing his cells dying without a virus, and he's saying, oh look, I don't need a virus for cells to die, when really he's just bad at cell culture. But there's no way to tell this for sure because he doesn't publish any of his results.
Lanka uses independent labs in Germany, and you can access leads to all of his work here on this website.
Let's keep going.
The next step, what they are doing, they use particles inside the tissue. They fix the tissue and cut it in thin slices, and look through the electron microscope. And they find some typical structures of dying cells, VLE, some tissue dots. They walk like amoebs. And when they cut through with the electron microscope, they show only the circuits and game those are viruses.
Cryo-electron microscopes can specifically identify viruses because they are powerful enough for us to see the actual structure and amino acid side chains of the proteins that make up the viral capsid. The sequence of these amino acids have to be coded for by a genome.
At this point, Dr. Lanka is simply saying this is how we have observed what we are calling viruses in the body, and Dr. Wilson is simply saying these particles that we are calling viruses, they are their own structures. They have been grown. They are not just willy-nilly random particles that are present. They have a structure to them. They were grown by some kind of order, some kind of organization, some kind of force, some intelligent force maybe we should say they are a construction. Dr. Wilson proceeds to educate us about the genomes of viruses and the genomes of the cells and how they are different, i.e. how the biochemical analyses are different between the two. How a virus didn't end up growing to be a cell because it has a different genome. That's essentially what he's saying. He's saying that if we take a part of the virus, it couldn't be found anywhere else other than in the virus. Therefore, viruses are real deal, proper. He's simply saying that because the amino acids are only made as a result of the virus's production, he's saying that's a reason that these viruses are self-replicating organisms. As if the cell can't grow this kind of package in some kind of effort to extricate certain materials from itself, to collect them together and extricate them out in a certain way, to break down into some building blocks, to break down in a certain way. Perhaps they will turn into larger units of life in certain conditions, as Lanka has stated. In terms of the cells of the human body, for example, they can't just go putting out particles into their environment. Any particles they need to put out need to work in with some kind of pattern for the rest of the cells that they are boarding with that make up the host, the whole human. So these cells will be extricating materials in a very specific way, tailored whatever works for this very complex organism.
Next time I see a fuck nigga and he popping it, I think that to me he popping it, I'ma pop pussy.
And the sequence of these amino acids in these proteins that we observe are specifically matched to the genome of the virus. Here's another example of a cryo-electron microscope taking a picture of a virus. This time it's HIV. On the left, you can see viral particles in all sorts of different orientations, but in each case, you can see that there's an envelope with contents on the inside. Those contents are the viral capsid. Here's another paper taking pictures of HIV, and you can see the structure of the viral capsid itself without the envelope.
I ain't got the sauce on it. You ain't got it, cause y'all washed up.
So just like how flat earthers deny that pictures of the earth exist, these virus deniers deny that pictures of viruses exist.
Actually, no, we just have a different interpretation of what exactly has been seen. The photographs of the Earth can only be interpreted one way. Some people say they are fabrications, they are made up, Photoshop. That is a vastly different dynamic to what is occurring here, Dr. Wilson.
10.3, they come up with another sort of particles, claiming where they say, oh, those are viral particles. And if you go into the technical details, you easily see that they centrifuge all the proteins in a test tube down to the button, and then they take it up with a syringe and mix it. And so the vortex is a mixture out of proteins, fatty acid, and detergents. And what we have soap bubbles.
That's right. He just said that viral particles.
Soap bubbles. First of all, that's not how people purify viruses in order to do cryo-electron microscopy. There are actually several ways that people prepare viruses for cryo-electron.
That's literally all we get for Stefan Lanka's point number 3 is Lanka saying that the centrifuged materials containing the virus end up with soap bubbles present. This doesn't mean anything, he's just taken this from what Dr. Lanka is saying and then just trying to dunk on him. There's nothing here. Lanka said something that sounds a bit silly from a certain perspective. Says many of these things.
Here's another paper taking electron microscopy pictures of measles virus. This is not a soap bubble. This is an envelope containing very specific viral capsids on the inside.
Actually, that material package called a measles virus could well and truly fit as a part of a soap bubble. If you don't know that, you need to go back to the drawing board.
Soap bubbles. Wow.
Fourth point. The fourth step, what they're doing, from billions of small genetic debris, they sequence it, they read the sequence of very small pieces, and then they add them up to a large piece. They add them up, and this process is called alignment. And it's incredible that they never ever carried out a control experiment with equally treated cell cultures, and just doing the same.
Yep, this is another very common claim by virus deniers that I've actually addressed before on this channel with my buddy Senstrand when we took down Andrew Kaufman. Basically, this is a total misunderstanding of how next-gen sequencing works. I'll put a link in the description to a very good video explanation of how next-gen sequencing works. But for this video, all you need to know is that it's a very, very large-scale version of the by-hand way that people use to sequence genomes. What you end up with at the end of all of these reactions are millions and millions of what we call reads. Each read does only cover a small segment of the genome, but each segment is covered by many individual repeats of that read, and these reads overlap with each other significantly. Since all of these reads came from one individual genome, it's not hard to put them back together like a giant puzzle, and they do only all fit together one way. And you can repeat this, and you don't recover the same viral genome from an uninfected cell culture.
Dr. Wilson, all of that which you have just said, Dr. Lanka teaches his students, anyone who learns from him. Just because you have narrated some features of the map does not mean Dr. Lanka is wrong.
If you don't believe me, you can use BLAST tools on NCBI's website in order to look for genes of viruses in host genomes. You won't find them.
Oh, are you saying we can go on some website and settle it right now and get to the bottom of it that Dr. Lanka is wrong?
And if you don't understand what I'm talking about, then you have a lot to learn before you deny virology.
Actually, I'm not convinced I have to learn any of that in order to rationally disbelieve in disease-causing viruses. If, in fact, that is what you mean by denying virology.
Oh, and by the way, here's a structure and image of another virus. This time it's hepatitis B.
Point 5. In order to build a genome of the virus, when they came up, I find a new virus in Wuhan, or I repeat the experiment, they always need a given genome as a point of orientation where they add the smaller pieces, where they do fit to.
This point's very much similar to the last point. They're discussing features of genomic maps, how we sequence genomes, how we find out what kind of genome different particles are that we've sampled. Dr. Wilson appears to be saying that what Dr. Lanka says is the case just can't be the case, and he seems to imagine that the way that he's telling the audience that it's just obvious that he's right. Here we've got two people, both PhDs in this relative field. They both have a lot to say about it. They both, metaphorically speaking, speak languages that general people like me even haven't learned, and they have a disagreement about how this map or that map works. Nothing Dr. Wilson has said to me equates to a reduction to absurdity of what Dr. Lanka has taught. It's actually quite absurd that Dr. Wilson seems to portray that it ought to be taken for granted that what he says equates to Dr. Stefan Lanka being wrong, being obviously wrong to his YouTube audience.
And as soon as I get hold of original sequence data, which was produced for HIV, I'm going to produce you coronavirus.
Oh my god. Okay, yeah, so this isn't one of his points, but I just had to include it. It's complete crazy town, and I love that he's actually claiming he can do this. So we'll all wait with bated breath while Stefan Lanka works on this experiment.
So let's come to point 6, Everybody thinks, and it was your question at the beginning, if that virus, you never ever saw a virus inside a living being or inside its liquid. And it's easy. Everybody can check it easily on every single photograph of a virus, which should show a virus. You'll find the notion that it's from a cell culture, but never from the blood, never from the salvia, never from the semen, never from another liquid of the body, never from a lymph node, never from inside.
Here are images of SARS-CoV-2 in a post-mortem patient tissue sample. Researchers of this paper were also able to track half of the virus through different human tissues by detecting the specific viral genome in many human organs, and also doing histopathology to look at the damage it was causing. So there you go, Lanka.
Stefan, welcome.
Take note of the date when this discussion was published on YouTube. It was before the date this paper that Dr. Wilson is sharing was published. Perhaps this was the first one of its kind.
I just showed you what you claim doesn't exist. And the reason that many papers that do cryology microscopy on viral particles took it from a cell culture is because they have to scale up in order to have enough viral particles in order to get a good high-resolution image of the virus.
And the reason Dr. Lanka has said this to people is because not that it refutes virology, not that it proves anything, but it's just painting the whole picture, a picture that general people don't have any idea about other than viruses cause disease. Dr. Lanka is simply sharing some information from the field of virology. He's not saying, look, all of these photos of viruses are from a cell culture and therefore virology is false. He's just saying, by the way, this is how it works. Dr. Wilson has wittingly pulled out a paper that shows what Dr. Lanka has said doesn't happen, albeit the paper was produced after Dr. Lanka said it, and albeit it doesn't really mean anything for the fundamental picture that we are contending with.
Researchers of this paper were also able to track half of the virus through different human tissues by detecting the specific viral genome in many human organs.
Actually, Dr. Wilson, I'd say that these researchers were actually just detecting different signal strengths that pertain to X particle throughout the different organs of the body, and they're just theorizing that they are tracking the path of the virus.
The 7th point is easy. In this review where I spoke about of the Max-Planck Institute on history of science, there you can read that when the old virology was disproving itself themselves, they find out when they took out animal experiments, they never ever were able to transmit the disease.
This is once again complete denial. But what better way to disprove this than by going through the example of SARS-CoV-2?
Lanka makes it clear. He is stating the reason the animals died when they did die is because they were treated harshly. They were damaged. For example, no one has ever caused an animal disease or killed it by introducing it to a microbe and had as a control during the experiment the introduction of a similar kind of chemical package, a protein package, to mock the introduction of the microbe.
at a similar rate, frequency, introduced in the same manner as the experimental microbe is being introduced. This is what Dr. Lanka is saying has not happened. In time we have harmed animals, the controls in the experiment do not account for the confounding factor of harming the animal through the means of which we introduce it to the microbe. So we might have an experiment where some monkeys were caused coughs when they had a virus sprayed down their lungs, but we won't have a control of the same kind of monkeys having a different kind of virus sprayed down their lung that theoretically doesn't infect those lung cells. And obviously spraying anything down a monkey's lung could cause it issues, most things at least.
In the description of this video, you'll find links to a list of several studies that together demonstrate that SARS-CoV-2 is a virus that causes COVID. This list includes studies where isolated virus was used to infect non-human primates and mice, and that infection led to disease in these animals.
Some monkeys had some of this chemical package sprayed down their lungs, dripped down their nose, and they had some issues. Not sure why. No one's kicking up a fuss that apparently SARS-CoV-2 can cause monkeys disease too. Apparently it was such a big deal when it mutated from a bat to be able to cause humans disease, and here we are causing monkeys disease. But rest assured, the introduction of this virus down these monkeys' nose or lungs caused them some issues. Wow. Replace the virus with concrete and we might have similar issues.
I talk about these studies more in depth in some of my other videos, but I promise you, if you send this list of studies to anybody who denies that SARS-CoV-2 is a real virus that causes COVID, they will have a meltdown. As if that wasn't enough for Lanka, though, I'm also including some studies where human challenge experiments were done with influenza virus. These are studies where human subjects are directly given influenza virus, and they get infected, they have a specific immune response, and they may develop disease.
Yeah, we have some experiments where humans had some influenza virus drip down their nose and they had some respiratory issues. They developed certain antibodies. Some of them developed fever. Some of them developed chills, shaking. Some of them got a higher dose than others. The higher the dose, generally, the more dis-ease could be measured in the human. The particles that we dripped down their nose or their lungs were detected more so in the people who had symptoms, i.e. when we dripped these particles down the person's nose, some of the people absorb them more than others, and these particles cause them issues. If they absorb them more than others, they're more likely to be detected with the particle than other people. Still, if we replace the virus particle with concrete particles and we have a special detecting mechanism for these concrete particles, we'll be able to spray concrete particles into people's lungs and we'll be able to measure the antibodies that generate. And the humans who absorbed the concrete dust more than the humans who had a thicker layer of mucus in their respiratory system, the humans who absorbed the concrete more, we're going to be able to detect concrete in them easier and more frequently. This is a good example of the situation currently. Here, 36 humans were challenged with SARS-CoV-2, had it intranasally administered drip down their nose. The humans who developed disease symptoms, they were the ones who were more likely to have been found with SARS-CoV-2 after the challenge. So if this SARS-CoV-2 is particle X, when we introduce it to these humans down their nose, the humans who are able to be found with it afterwards are more likely to have endured some kind of disease, which is to say the humans who absorbed the particle, they were more likely to have some issues as a result of absorbing that particle. And I say they absorbed the particle because they were the ones who were able to be found with the particle. So if we release a bit of gold in them and they happen to absorb the gold in their cellular network, they lodged it and other humans didn't. If we have a gold detector, we're going to be able to detect the gold in the humans who had the gold lodge in them, have absorbed it. And if the gold, when lodged in these cells of the respiratory system, if it causes issues, then the humans who lodged more of it in their respiratory system are going to have more issues. And these are the humans who are going to have to extricate that gold through feverish processes. They could find the particle that they dripped down their nose on average for 10 days afterwards, after dripping it down their nose. They could find it here and they could find it there, different parts of different liquids of the body. Imagine the body is sprayed with this particle, it absorbs all these particles, different bodies are going to react differently to it, and like I said, different bodies are going to have different levels of mucus sitting on their cells, which will take the brunt of the particles. The bodies without so much mucus, these particles might just lodge straight in those cells, and it might be a very uncomfortable experience for those cells that never accounted for this kind of situation to occur in such a fashion. All of these particles landing down the respiratory system and no control whatsoever, such as another protein package or fat acid package. Dr. Wilson, you've been roasted. If you want a free copy of my book, I'll send it to you via email.